Hey guys, my name is Max and welcome to Simple Biology. Today we're going to start talking about Mendelian genetics, and we're going to start with some of the less complex concepts, because from here we can then build on to the more complex concepts. Now, Mendelian genetics was named for this guy named Gregor Mendel, who was a monk slash pea farmer. Basically, a monk for his time was like being a philosopher was in the time of the Greeks. He was a big deal. He set the framework for our modern conception of genetics, and that's why his ideas and the results of his tests and experiments are still important today. We're going to start talking about just the simple concepts that he helped us gain. First, we're going to start by talking about dominant and recessive traits. Now, most organisms that we encounter will have two copies of each gene. Humans have this, mice have this, every single animal has this, and most eukaryotes also do. What this means is that both of these copies of these genes, both copies of the gene, will be playing a role in the actual outcome of your appearance, of your structure and your function. Now, a definition we'll need to know is an allele. Now, an allele is one specific form of a gene. There are often more than one form of the gene. For example, you could have an allele for white hair color, you could have an allele for black hair color, for brown hair color, for blonde hair color. As many as you want. Generally it will be two. For the purposes of our studies, it will almost always be two, and you can assume that unless it tells you otherwise. Also, often these pairs of alleles will interact with each other in a very predictable way. One will be dominant and one will be recessive. In other words, if there is a dominant allele of a gene, it will have the phenotype, or the organism will have the phenotype of that dominant gene. It will completely hide any recessive traits that will be given by the recessive allele. Because of this, there are three possible genotypes. I'll define a genotype in a minute. Essentially, when you see these symbols, the capital letter will be a dominant allele, the lowercase letter will be a recessive allele, and you can either have two dominant alleles, one of each, or two recessive alleles. And for all intents and purposes, both of these two sets of alleles will look identical from the outside. Now, before we go on, let's identify some key terms. Now, homozygous means that both of the alleles are the same. Homo means same, and zygous will be the other part of the word. So the other way you can have it would be heterozygous, and this means that the organism actually has two different alleles for the same gene. If you remember back in the last slide, I showed you that you can either have two capitals, a capital and lowercase, or two lowercases. The first and last are both homozygous because both alleles are the same, and that middle one, where you have a capital and a lowercase, or a dominant and a recessive allele, is heterozygous because the alleles are different. Now, a genotype, I mentioned this phrase in the last slide. A genotype is essentially the alleles an organism has the genes for. So, this is if you look at the genes of the organism, it's what that is. So, let's make more sense when I explain the other form. So, a phenotype is rather than being the genetic description of the organism, it's the form or function is expressed as a result of the genotype. So, a phenotype will show you what the organism looks like, how it acts, how it behaves. Whereas the genotype will be its actual pair of genes. It's what it has the genes that code for. So you can see that sometimes more than one genotype will result in the same phenotype, and that makes things incredibly easier to work with. Now, next we're going to look at Punnett squares. Now, Punnett squares you've probably seen them before, and they can get really boring. They can get really frustrating. Not frustrating, but it's just kind of tiring, because there are really only 6 possible combinations with simple Punnett squares, and we're going to look at those. A Punnett square is a way of testing the breeding of 2 different organisms of the same species just for a single gene, and looking at the statistically probable outcome of their breeding, of what their offspring will look like. So essentially, you set up a square with this sort of grid-like pattern, and you will split its genotype into 2 different sections of squares. Now for this, it would be a homozygous dominant, meaning both of its alleles are the same and it's dominant. So capital W, capital W. So on either side you'll have capital W's, and this will actually be a cross between 2 of those. So as you can see, every single organism will have this, or every single organism that's an offspring will have this same genotype where it's homozygous dominant. Next, you can have a homozygous dominant with a heterozygous, and the result of this cross will actually be a set of organisms that all has the same phenotype, but half of which have that recessive allele still in them. Now, what we call an organism that shows the dominant phenotype but still has that recessive allele is we call it a carrier. And you may hear this a lot with like genetic diseases that can be passed on if 2 carriers interbreed. We'll get into a Punnett square for that sort of situation later. But essentially what happens is the carrier doesn't actually have that trait, but their kids could possibly have that trait because the allele for the trait is in their genome, or is in their genotype. Now you also remember that every single one of these had the same phenotype. What we'll see is that if either of the parents is a homozygous dominant, then every single offspring will have the same phenotype for that gene as that homozygous dominant parent. We call this a true breeder or a pure breed. And so because it's a true breeder, all of its offspring will have that same phenotype as it. So that's another term that's going to be good to know. Next, we have a cross between a homozygous dominant and a homozygous recessive, and you'll see that every single offspring will be heterozygous, or a carrier, as we established. Over here we have a cross between two heterozygous organisms, and this is going to be where we're going to look at the most. This is called a monohybrid cross because both of them are hybrids. A hybrid is another term for a heterozygous individual for a certain gene, and mono is because we're only looking at one gene. We're not looking at multiple genes. That's where stuff gets more complicated. So this is a monohybrid cross, and you see that the ratio for genotype is 1 to 2 to 1. This is the only cross that will have all 3 genotypes, and that's actually very significant because this is used a lot when we have practice problems in genetics, and especially with high school and college level biology. In addition to this, a lot of times you'll have questions that will ask, you'll start with this Punnett square, and so you'll have a cross between these 2 organisms, and all the results will be heterozygous. Then they'll take the results of that. They'll take those offspring, breed them with each other, and you'll have this test cross. A lot of people get tripped up in that because you went through 2 different breedings, and in the end, it'll be the 1 to 2 to 1 ratio, which will be very good to remember. Next, we have a cross between a heterozygous individual and a homozygous recessive individual. And what we see here is that half of the offspring will be heterozygous, and half the offspring will be homozygous recessive. This is interesting because that's the same proportions as their parents. This is one of the few that will keep that without both of the parents being identical. This type of cross is what's known as a test cross. When you take that homozygous recessive individual and breed it with another organism, you can tell whether the other organism is homozygous dominant in the case of this cross, or heterozygous, as in the case of this cross. And so this specific cross is like a positive result of a test cross. It's basically testing whether the organism is a carrier or homozygous dominant. The last cross will be between two homozygous recessive individuals, and as you would expect, all their offspring are homozygous recessive. The last Punnett square you're going to need to know is a little bit more complicated. In the last slide I mentioned a monohybrid cross. This is what's known as a dihybrid cross. This is essentially the cross between two organisms which are both heterozygous for two separate genes. That's why it's di instead of mono, because there are two genes involved now. So right here we have the genotypes of each of the organisms that are parents, and you can see they're heterozygous for two different genes. Here we split them into we split each of the two genes into their separate alleles, and these are the four possible gametes that they can have. These are the four possible combinations of alleles that they can pass on to their offspring. As we do the cross, we see that we get this interesting looking result, and what you need to remember is that the phenotypic ratio is 9:3 to 3:1. Also be able to replicate this if you need to. You just take whatever is on the top and the side for that certain cell and just add the set of letters together. So 2 capital W's, 2 capital H's, put them all together, capital W, capital W, capital H, capital H. You will not need to know the genotypic ratio, I hope, because it's incredibly complicated and it's just useless, really. But that phenotypic ratio, if you see that 9:3 to 3:1 ratio, know that it is a dihybrid cross. That's it for now. We'll be getting on to some more complex Mendelian genetics in our next video, but for now, it's as simple as that.